Hello, my name is Dr. Richard Thwaites, and I'm the Medical Director of Falcon Physician Reviews. I'd like to take this opportunity to introduce you to our biochemistry modules for Level 1 of the COMLEX and Step 1 of the USMLE. Biochemistry is one of those subjects that often causes students a great deal of stress. And one of the reasons for that is there's actually two types of biochemistry. There's the PhD level biochemistry, which enforces every step in every pathway. And there's clinical biochemistry, which enforces the steps that break or the things that go wrong and manifest themselves in clinical presentations. The USMLE and the COMLEX tests clinical biochemistry. And that's why it gives me a great deal of pleasure to introduce Dr. Adam Sigmiller. Dr. Sigmiller has a PhD in biochemistry, but he also has an MD, and as such has taken the USMLE. Because of this, he's going to be able to help you focus on clinical biochemistry as is tested on the USMLE and the COMLEX. Now, without any further hesitation, let's begin our first biochemistry module.
The section on molecular biology is centered around what we call the central dogma of molecular biology. The central dogma of molecular biology is the theory by which we connect the genotype of an individual, that is, the contents of their DNA in each of the cells in their body, to their phenotype, which is the expression of that genotype in the function and the appearance of that person. The central dogma of molecular biology says that the genetic information in each and every cell is contained in, transmitted through, and decoded to protein via nucleic acids. And it's in that protein, then, that we see the phenotype in individuals. The conversion of DNA, the division of DNA from one cell into two cells is called replication. And that would be the first step. That's how DNA is transmitted to DNA, how genetic information is transmitted from a parent cell to two daughter cells, or from one organism to its offspring. The process by which DNA gets decoded into RNA is called transcription. The process then by which the RNA is translated into proteins is called translation. And in the next series of this lecture, we will deal with each of those processes in detail. First of all, we have to understand nucleic acid structure, however. Nucleic acids are composed of nucleotides, and each of these nucleotides has three distinct parts. The first is a pentose sugar, that is a 5-carbon sugar. This is either ribose, which we see in RNA, ribonucleic acid, or deoxyribose, which we see in deoxyribonucleic acid, or DNA. Attached to that pentose sugar is a nitrogenous base. This base is either adenine, guanine, cytosine, thymine, or uracil. RNA and DNA have adenine, guanine, and cytosine in common. Thymine is only seen in DNA, and uracil is only seen in RNA. In addition, there are one or more phosphate groups which are attached to the pentose sugar. The structure of each nucleic acid looks something like this, then. You have the pentose sugar at the base, and attached to one of the carbons you have a nitrogenous base, which shown here is cytosine, and attached to the last carbon we have a phosphate. Now, both DNA and RNA have a similar structure. Both of them are polymers. In other words, they are long chains of these nucleotides covalently bonded to one another. The covalent bond is a phosphodiester bond between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another. Now, at this point, we should say something about the nomenclature of the carbons on these nucleotides. The first carbon that we number carbon number 1 is the one that's attached to the nitrogenous base. Then we go around the ring and number them carbon 2, carbon 3, on which this phosphodiester bond is linked, carbon 4, and then carbon 5, which the phosphate is attached to on the other side. Polymerization of these, that is the process by which one nucleotide is added to the previous nucleotide, always occurs in the 5' to 3' direction. That is, the 5' phosphate of the free nucleotide is added to the 3' hydroxyl group of the growing polymer. Now, RNA is slightly different in DNA. The only structural difference between the two is what is attached to the number 2 carbon. In DNA, that's simply a hydrogen, but in RNA, there is a hydroxyl group attached to that position. In addition, as I mentioned before, we have a uracil group which is substituted for thymine, the base that we see in DNA. Now, the bases of DNA and RNA can pair to each other, can bond to each other through specific hydrogen bonds. This is the process that we call base pairing. Now, in every case, both in DNA and RNA, the adenines are hydrogen bonded to either thymine in DNA or uracil in RNA by 2 hydrogen bonds. Similar to that, the guanine is hydrogen bonded to a cytosine by 3 hydrogen bonds. Now, this difference between these 2 different types of base pair is important because it indicates to us that the bond between guanine and cytosine is stronger and more stable than that between adenine and thymine or uracil. Now, the nucleic acids then, because they have this ability to base pair, can interact with each other to form double helices. And in this structure, what we see is a thymine hydrogen bonded to an adenine and a cytosine hydrogen bonded to a guanine. The result of that is something that is an anti-parallel and complementary structure, and it forms the double helix that we're familiar with in DNA. These 2 properties are very important in trying to understand how DNA interacts, the strands of DNA interact with each other. It's important to remember that these 2 strands are always anti-parallel. That is, they go in opposite directions from 5' to 3', and they're always complementary to one another. That is, G's are always bonded to C's, and A's are always bonded to T's as it forms the double helix. Now, RNA can make these kinds of structures as well, but RNA is almost always single-stranded. You rarely see RNA in a double-stranded conformation. So the base pairing that occurs in RNA molecules tends to be intramolecular. That is, base pairs from a strand of RNA binding and forming hydrogen bonds with base pairs in that same strand of RNA is indicated in the illustration here. In DNA, these bonds are intermolecular, meaning it's hydrogen bonds between nucleotides of different strands of nucleic acids. Now, there are approximately 6 × 10⁹ base pairs of nucleotides in any mammalian cell. And that's a problem because if you stretched out these nucleotides on end, they would stretch out to about 2 meters in length. Obviously, that's not going to fit in the nucleus of any cell that we know about. In fact, most cell nuclei are only about 10 micrometers in diameter. And so obviously we've got to compact that DNA, we've got to organize it, and we've got to condense it somehow in order to get all of those 6 × 10⁹, 6 billion base pairs into a single nucleus. The primary organization of DNA, the first step in forming this compact structure, is called a nucleosome. This consists of DNA wrapped around some specialized proteins which are called histones. Histones are proteins which are enriched in basic or positively charged amino acids. These are particularly arginine and lysine. We'll talk about that in a different lecture. But the important thing to realize here is that this allows these histone proteins to bind to negatively charged DNA. Now, the DNA binds about approximately 146 base pair, wraps around each octamer of histones. Now, that octamer consists of 2 molecules each of histone H2A, H2B, H3, and H4. In between these histone DNA structures, there are approximately 55 base pairs in a sort of spacer in between. Now, this gives the nucleosome an appearance of beads on a string, as you can see in the illustration which is shown here. Now, histone H1, which binds to that 55 base pair spacer, further organizes these nucleosomes into a polynucleosome or a nucleofilament, similar to what you see to the right of this figure here. So the DNA wraps around the histones to form nucleosomes, and then groups of nucleosomes form a polynucleosome or a nucleofilament. And this allows the DNA to be compacted and condensed so that it can fit into this small 10 micrometer nucleus. Now, in the nucleus itself, DNA is found in two different states, euchromatin and heterochromatin. Euchromatin is less condensed than heterochromatin is, and this allows the transcription machinery to have access to the DNA so that it can be actively transcribed. So euchromosome represents those genes which are actively being transcribed and forming RNA, which goes on to form protein. The heterochromatin, by contrast, is more condensed. This is the DNA which is transcriptionally inactive, that it is not currently being transcribed. If you look at a picture of a nucleus in a scanning or a transmission electron micrograph, which you see at the right here, you can see that the heterochromatin is the denser appearing DNA in the nucleus, whereas the euchromatin is the more open, less condensed DNA. This ends the lecture on DNA.